Hi, this is Erica Henry, and I am doing my advanced concepts presentation on fluoruracil. Okay, so we need to know basically what fluoruracil is. It's one of the most commonly used drugs to treat different types of cancers, and it's usually used in a combination or a cocktail. It's a group of chemotherapy drugs known as antimetabolites. And these antimetabolites stop the cancer cells from spreading, working properly. Let's see. Whoops, sorry. And then fluoruracil, also known as 5-FU, is a cell cycle inhibitor, and it specifically inhibits at the S phase. And chemically, it's an analog of a uracil by integrating into the DNA or the RNA. And then this induces the cytotoxicity of the cells, which eventually leads to the cell death. Okay, back down here. How is it metabolized and what does it inhibit? So it metabolizes, it is metabolized in the liver, as is most medications. It's broken down by an enzyme called dihydropyrimidine dehydrogenase. And, ooh, goodness. And it interferes with the nucleoside metabolism, and it then can inject itself into the RNA, into the DNA, which, as I just spoke about, causes the toxicity and the death of the cell. It inhibits the thymidylate synthase, also called TS, which is an enzyme that catalyzes the conversion of deoxyuridine monophosphate to deoxythymidine monophosphate. And so all of these acronyms are going to be used, or abbreviations are going to be used when we get to the last slide, where I kind of show you the mechanism of how this works. So it's really important to try and keep them straight, which has been a challenge for me. Okay, so how does it inhibit RNA synthesis? It works by inhibiting the process of what's called pre-RNA to mature RNA, and it disrupts the post-transcriptional modification of the transcriptional RNA and the assembly activity of the snRNA and protein complexes. So this inhibits the splicing of the pre-mRNA, and this is important because the balance between the non-cancerous isoforms and the cancerous isoforms is a big contributor to the growth of cancer and the beginning of it. Okay, so here's the mechanism. And I'm tried really hard to try and understand this. But basically, you've got the 5-FU, which is the fluoruracil. And the 5 here means that there is a fluorine atom on the 5th hydrogen of the compound. So that's where the 5 comes in. So if it enters the cell, and then it can be broken down into, 5-fluorouridine ribonucleotide metabolite, which is what this is. And then it can go, so 5-FU, if you follow this down, does the damage to the RNA. Now, this is the analog FUDR, which stands for 5-fluoro-2-deoxyuridine 5-phosphate. And so if you follow this down, this will do the damage to the DNA. And eventually this leads to the apoptosis of cancerous cells. So you've got fluorouridine monophosphate, the diphosphate, and the triphosphate. So all of these get broken down, and then it causes the damage to the RNA. Same goes over here. It breaks it down, so you've got your monophosphate, your diphosphates, your triphosphates. And then here's that TS that we talked about earlier, the thymidylate synthetase. So yeah, a lot of words, and I'm not quite sure I understand it totally, but that's kind of what's happening. So the precise mechanism of action actually hasn't been totally fully determined by scientists, but the main thought is that the mechanism of the fluoruracil is thought to be the binding of the deoxyribonucleotide of the drug FDUMP, so right here, and the folate cofactor N510 methylenetra tetrahydrofolate to the TS, and to the TS is a covalently bonded ternary complex, which results in the inhibition of the formation of thymidylate from uracil, which leads to the inhibition of DNA and RNA synthesis and cell death. Fluorouracil can also be incorporated into RNA in place of the uridine triphosphate, producing a fraudulent RNA and interfering with the RNA processing and protein synthesis. So that's just kind of a better description of what's going on, but I hope that was helpful. If you have any questions, let me know. Thanks.